With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello students, in this question there is some data given. In this case it is given that a sample of enzyme, the name of the enzyme is lactase, and it was isolated from the intestinal lining of the calf. Now assays were undertaken to evaluate the activity of the enzyme sample. So the substrate for the lactase is the disaccharide lactose, and when lactase enzyme acts on this lactose, it breaks it down into two molecules. Now one of the molecule is the glucose molecule, which is a monosaccharide, and another molecule is the galactose molecule, which is also a monosaccharide. So let's see the first assay. There is lactose concentration which is given. There is concentration of the enzyme sample which varies according to the time period, that is the reaction velocity will also change accordingly. So in the first assay it is given that if we keep the lactose concentration same, then what if we change the concentration of the enzyme sample, what will happen to the reaction rate? So reaction rate is given here. So this is how with the change in the enzyme concentration the reaction rate varies. While coming on to the second assay, in this case it is given that the lactose concentration will change. See here, these are the variables, this is changing. Whereas if we keep the enzyme concentration same, then how will the reaction rate vary? So in this case the enzyme is not variable. So that means we can say that in case of assay number 1, that is the test 1, the variables are, we can see that the enzyme sample values are varying here. So in this case we see that this is the concentration of the enzyme sample, right? Whereas in case of assay 2, what is varying? It is the lactose concentration, that means the substrate is varying. So lactose concentration is varying. So that means the first assay tells us about the effect of increase in the enzyme sample, whereas the second one will help us to know that that the concentration of the lactose sample increases. So in that case, this option which says that this is concentration of the enzyme sample and the lactose concentration. So this becomes the right option for this question, right? So in assay number 1, enzyme sample. In assay number 2, the substrate sample is changing, and accordingly we are checking that how it affects the rate of the reaction in this case. So this becomes the right option for this question. So this is all for this query. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.